So we're ready. All right. Good morning, everyone. Thanks a lot for either coming here or joining online. There is quite a few people online. And it is my pleasure to introduce Saranya Pullanchery. Is that more or less?
Yes, that's correct.
Okay. So my last name is complicated too. So I know how it feels when they say completely wrong. All right. So Saranya, in a few words, did her undergrad in the Aitzol Institute in India. Then she moved to the US for her PhD, and she developed it at Penn State University. And nowadays she's a postdoctoral researcher at EPFL under the supervision of Sylvie Roque. And today she will be talking about shining light on the interface. And I'm gonna leave the details for her, of course. And so that's all I have to say. Saranya, the floor is yours.
All right. Thank you, Aitzol. And hello, everyone. So today I'm really excited to share with you two stories about shining light on the interface. So interfaces are everywhere in nature, right? Whether we are studying chemistry, physics, or biology, we often come across many examples where two media come into contact with each other and you generate a boundary layer. So at the boundary layer, the molecular structure or the interfacial potential is going to be very different from either of the two media. So what is the molecular structure of an interfacial layer? And how do different molecules interact with each other in this thin layer? And how those interactions then determine the macroscopic properties of the system? To answer these fundamental questions, we really need to be able to separate this first few molecular layers from either of the bulk media. And this is a challenging task. And ideally, we would like to do that without using ultra-high vacuum techniques. So in the next few minutes, I would like to show you two separate problems where we addressed this type of challenges. So the first one is about metallomembrane interfaces. So you might have heard about metalloproteins, and those are studied, more well studied, and they're known for such a long time. But about 10 years ago or so, we actually found out that specific divalent cations, especially transition metal ions, can form specific tight complexes with negatively charged lipids in the membrane. And we call them metallomembranes. So if we look at the common metal ions that are present in our body, ions like calcium and magnesium that are present at millimolar concentrations, these ions have been known to bind to negatively charged lipids. And binding of calcium can cause the fusion of negatively charged lipid membranes. It can change the phase transition temperature and cause change in the phase behavior of the lipids. It can also trigger protein signaling cascades. Now, if you go down this lipid, ions like copper or zinc, the transition metal ions, have been studied to much less extent in this context. And in this particular case, we were interested to study the interaction of zinc ions with negatively charged lipids. So why zinc, right? Zinc is interesting because it's the second most abundant transition metal. And zinc can also induce vesicle fusion like calcium, but it does that at much lower concentrations. And zinc is also very important in the brain because even though the overall free zinc concentration in our body is quite low, the concentration of zinc can reach millimolar levels in certain part of the brain, for example, in synaptic vesicles. And zinc 2+ ions, therefore, play a very important role in neurotransmission. So because of these reasons, we wanted to look at how zinc interacts with this negatively charged lipid called phosphatidylserine, or PS. And so to do that, first we wanted to look at lipid membranes. So we used fluorescence microscopy. So here you can see that we form a supported lipid bilayer. So this is a lipid bilayer on a glass substrate. And we add a small percent of fluorophore labeled lipid so that you can visualize these lipid membranes. And you have PS, which is the negatively charged lipids, and PC, which are the zwitterionic lipids in the membrane. And you see a uniformly fluorescing bilayer in the absence of zinc. But when you introduce 100 micromolar zinc, you start seeing these bright fluorescent spots that form and grow. So at this point, we also found out that these are actually not in the plane of the lipid membrane. They're actually three-dimensional structures that grow out. So the question was that, well, is this phenomenon specific to zinc? Can other divalent cations also induce such structural changes? Very interestingly, it turns out that it's a pretty much a very ion-specific phenomenon. Zinc is the only ion that can form these blebs very efficiently. Calcium ions can also do the same phenomenon, but much less effective. But ions like magnesium, copper, and nickel that we tested, they do not form blebs in the lipid membrane. So why is that, right? What is special about zinc interacting with PS lipids? So to study this, we have to really look at that interfacial layer, the first 1 nanometer or so that contains the lipid head groups, the ions, and the water. So to study this interface, we use this technique called vibrational some frequency generation. And the way we do that is by forming a monolayer of lipids at the air-water interface. And these are called Langmuir monolayers. You have 2 barriers on either side. You compress the barriers. You increase the packing density of lipids in the monolayer. So just to give you an idea how the system compared to the lipid bilayer that I showed you just before, if you compress the monolayer to 30 millinewtons per meter, that is equivalent to the internal pressure of a lipid bilayer. So that's how we can compare these two systems. And once you form this monolayer, we then overlap 2 ultrafast beams. You have an infrared laser beam and a visible laser beam. And you spatially and temporally overlap these 2 beams on the surface. And the visible frequency in this experiment is usually kept fixed. But the infrared, we can tune it to any frequency in the mid-IR range that we want. And whenever this infrared is in resonance with any of the molecular vibrations that are present at the interface, it could be the lipids or it could be the water, you generate a 3rd photon that comes out of the interface, and that's a some frequency photon. And this some frequency photon only is generated if you have the IR is in resonance, right? So you have a resonant process here. Now, what is really special about SFG technique is that it's inherently interface selective. And this is because this is a 2nd order nonlinear process. And due to the symmetry selection rules, for any molecular vibration to be SFG active, it has to be present in a medium without inversion symmetry. So we can take water as an example. So imagine water molecules in the bulk. They are sitting in a centrosymmetric medium. So all the dipole contributions cancel each other. So they do not generate an SFG, but water molecules at the interface, or for that matter, any molecules at the interface between 2 media have to necessarily break inversion symmetry. So they are the only ones that will generate SFG photons. So we really use this interface selectivity to look at the SFG response as a function of IR frequency. So now let's see what happens, right, when you add zinc to a PS lipid monolayer. So if you form a PS lipid monolayer in the absence of any ions in the subphase, just lipids and water, you see a spectrum like this. So this is the SFG spectrum. You can essentially interpret it similar to infrared or Raman spectra. So there are these sharp peak below 3000 wave numbers, and these are from the lipid tails of the lipid monolayer. So they are highly ordered. They're highly ordered at the interface, so then they generate very strong signal. And then what's really interesting is this broad region between 3000 and 3800 wave numbers. What we need to remember here is that these are water molecules just in the interfacial layer, right? This does not involve the bulk water. So we are specifically looking at interfacial water. And I've marked here 2 types of water. So there are some water molecules that are directly interacting with the lipid head group, the very first layer of water. Now, the very first layer must have a somewhat broken hydrogen bonding network compared to water molecules that are further down into the bulk. So those weakly hydrogen bonded water molecules would show up at higher frequencies. And then water molecules that are in the subsequent layers, they can also be ordered, or they can also be in a broken inversion symmetry environment. Why? Because we have a negative charge at the surface, right? So those negative charges are going to create an interfacial electric field. That electric field then orders water dipoles. So those molecules will also be detected by SFG. So we can see those water molecules, since they are in a more strongly hydrogen bonded environment, they will show up at lower frequencies. So now what we can do is that we can now use this interfacial water as a probe for our metal lipid interaction. So you can see if I add zinc to the subphase, I see an overall decrease in the interfacial water peak. And this happens because when zinc binds to the lipid head groups, it neutralizes the charge, and that results in a decrease of overall interfacial electric field, or you displace some of the head group waters, and you see an overall decrease here. And we were then able to show that you can quantify the strength of the zinc PS interaction by monitoring this intensity or the amplitude of this 3200 water peak. And it turns out that the dissociation constant for this interaction is around 260 micromolar. It's 2 or 3 times tighter than the binding of calcium to PS. So that's good. We can characterize how strong this interaction is using water peaks. Now the question is, where exactly does zinc bind on the lipid head group? To find that, we can look at the vibrational peaks on the lipid head groups. So for example, here is a phosphate, and the phosphate symmetric stretch vibrates around 1100 wave numbers in the IR spectrum. So we do the same SFG experiment around this frequency region. And here I have shown one water molecule's hydrogen bonding to the phosphate, but in fact, there are many. The phosphate is well hydrated. If it's well hydrated, it vibrates around 1100. And what's really good about looking at this peak is that the peak is highly sensitive to the hydrogen bonding environment around it. If the hydrogen bonding environment gets weaker, the peak shifts to higher frequencies. If the hydrogen bonding environment gets stronger, the peak will shift to lower frequencies. So if the metal ion can come and bind in such a way that it dehydrates the phosphate and forms a contact pair, then that should shift the frequency of the phosphate peak. And that is exactly what we see. We see that when zinc binds, it actually shifts the phosphate symmetric stretch vibration by around 11 wave numbers, which tells us that it actually binds in a way that it forms a contact ion pair. We then similarly looked at the carboxylate vibration on the head group. And we again here saw that there is an increase in a high frequency population. So that gives us evidence that zinc also interacts with phosphate and carboxylate. So at this point, we also had some theoretical support from DFT simulations, and we were able to come up with a possible complex that is formed between zinc and 2 PS lipid head groups by interacting with the phosphate and carboxylate. So this is all good, right? It's showing us that zinc binds and neutralizes the charge on the lipid membrane. Now the question is, how does the monolayer structure, and when I say structure, I mean the phase behavior of the lipid monolayer change. To do that, we can do measurements that are called as pi-A diagrams, or they're essentially surface pressure area measurement. These are thermodynamic measurements. You form a monolayer at the air-water interface, you compress the monolayer, and as you are compressing, you measure the surface pressure in the metal. And if you look at this black pi-A diagram here, this is in the absence of any ions. You can see that the monolayer exists as a single phase. But when you add zinc, you see 2 effects, right? The first one is that the isotherm shifts to lower area per molecules. What that means is that when zinc binds to 2 lipid head groups, it brings them together, and this results in an overall reduced average area. And the second is that we see a phase transition right around 30 millinewtons per meter. So now if I can also visualize this by adding a little bit of fluorophore labeled lipid on the monolayer and then taking the trough under a microscope. And if I do that, I see that initially in the absence of zinc, I have a fairly uniform monolayer, but zinc induces the formation of these ordered domains in the monolayer. So the dark regions that you see are the regions where the dye is excluded from. So it really creates these circular ordered domains on the monolayer. So at this point, we can also try to characterize what's going on in the monolayer phase behavior using spectroscopy, because the selection rule of some frequency generation that I described to you earlier really makes it a good technique to detect this kind of order disorder changes of the monolayers. Because if you have, imagine you have a monolayer with a lot of gauche defects, and these CH2 groups in the middle will be in a medium with broken inversion symmetry. So they will show up in the spectrum, and you have CH2 groups, and the terminal CH3 groups are anyway pointing towards air, so they also show up in the spectrum. But now if you have a very well-ordered monolayer, the CH2 groups in the middle are sitting in a medium with net inversion symmetry, so they all cancel each other. You only detect the terminal CH3. So by using these two peaks, if they're present or absent, or using the ratio between them, we can quantify the extent of order or disorder in the monolayer. So this is what we did. We first look at the monolayer in the absence of zinc, and we see that the CH2, when you increase the surface pressure from 15 to 45 millinewtons per meter, the CH2 group goes down and the CH3 goes up. If you quantify the ratio of amplitudes, it pretty much monotonously increases, which shows the monolayer getting more ordered. But if you now add zinc and do the same experiment, you see a very sharp increase in that ratio, right around 30 millinewtons per meter. So this is the same pressure where we saw the phase transition in the thermodynamic measurements, right? So this now ties it all together and tells us that zinc binds, it neutralizes a charge, it brings the lipid head groups together, and you see a phase transition in the monolayer. Then the question is, what is really special about zinc, right? And we did the pi-A diagrams with all the other divalent cations that we were interested in. And interestingly, all of them can induce monolayer phase transitions. But zinc is the only ion that induces phase transition around 30 millinewtons per meter or slightly below. So that's what is different about zinc. So now I have drawn a line here, right? This line, if you remember what I said in the beginning, 30 millinewtons per meter is also the internal pressure of a lipid bilayer. So it makes sense that zinc is the only ion that's able to do this phase transition below that threshold. Now, that's not the only case, that's not the only reason why zinc is unique. We then looked at this phosphate stretch that I showed you before, and the phosphate stretch shifts only in the presence of zinc or calcium ions. That means that zinc and calcium ions are able to bind to the phosphate in a manner that dehydrates the phosphate and forms contact ion pairs. But the other ions, copper and magnesium, even if they do interact with the phosphate, they're not dehydrating the phosphate group. So they're forming probably solvent-separated ion pairs. So that tells us that not just this phase transition that you see in the monolayer, but a contact ion pair formation is also a necessary condition to see the blips in lipid membrane. So we can use all this information that we got from the surface-specific spectroscopy and now go back to the initial observation that I showed you, right? So we had a lipid bilayer, we had PS lipids on both inside and out, so the inner and the outer leaflet, but you introduce zinc from the top solution. So you essentially bring the lipid head groups on the top close together. So that means you have a net reduction of the area of the top leaflet. And because you have this area mismatch, the membrane will have to then bend outward to accommodate for that area difference. So this is what we think is going on in this formation of these blips. So hopefully I've shown you that for studying planar interfaces, SFG is a really nice technique. We can get a lot of detailed information about the fundamental mechanisms of processes that are happening. So now, but in reality, most of the interfaces in nature are not planar, right? Most of the interfaces are curved. So in the second part, I want to show you an example of how we looked at a curved interface. And in this part, we are going to talk about oil and water. So you all know that oil and water do not mix, right? And this is called the hydrophobic effect. So if you look around, the hydrophobic effect is everywhere, from the cleaning action of detergent to cheese making to protein folding inside cells, right? A single phenomenon is responsible for such a wide range of processes. But the molecular mechanism of hydrophobic effect has been very controversial. And one of the controversies is that even though you cannot mix oil and water, you can form oil nanodroplets in water without adding any surfactants. And this is a very surprising observation, right? So here I have an example. These are hexadecane droplets in water. It looks like this. And I measured the dynamic light scattering size distribution with DLS. And after preparation, after 18 days, it looks more or less the same. So it's very surprising. Why are these emulsions stable, or they're not stable, they're kinetically stable in the absence of any stabilizing molecules? And interestingly, what people observed, in fact, this is a very century-old observation, that if you apply an electric field across this emulsion, the droplets always move towards the positive electrode. And this is what we call as electrophoretic mobility, which can be converted to a zeta potential value. So you measure how fast the droplets are moving, then you use hydrodynamic models to measure the zeta potential. So hexadecane droplets in pure water have a zeta potential of minus 56 millivolts. So, okay, they're negatively charged. That gives an explanation for why they're stable for such a long time. But then the question is, why are they negatively charged? Because we just have oil and water, right? And this question has been around for such a long time. So people did zeta potential measurements for a variety of droplet systems, not just oil and water, but also bubbles. So this is a compilation of data from over the years on different type of oil droplets and air bubbles. And you always see this consistently, this negative zeta potential around neutral pH. And if you go from acidic pH to basic pH, you see that the zeta potential keeps getting more and more negative. So based on this data, one of the obvious explanations, or probably a naive but obvious explanation, is that you go to high pH, you have more negative zeta potential. So it must be that hydroxide ions go to the surface, and that makes it negatively charged. So, and then at neutral pH, how can you explain that? Then it's probably that you have autoionization of water, you have some amount of hydroxide that's giving rise to the negative charge. But again, this type of explanations were made when we had no surface-specific spectroscopies available, but you can always do surface tension measurement and surface spectroscopies to see if hydroxide is really present at the surface. So I want to show you two examples of that. So this was a surface tension measurements made by James Beattie in 2014. So they looked at air-water interface, which is again a hydrophobic water interface surface tension. And you can see that when you make the pH go from, let's say, pH 2 to pH 10, the surface tension is more or less the same. But if you go to really acidic pH, the surface tension goes down. Okay, so that could mean you have protons going to the surface. It's still debated, heavily debated, but look at the hydroxide data. Only at very high pH, you have an increase in surface tension, which could potentially mean that it's being excluded from the surface. And then we also have some data from X-ray photoelectron spectroscopy, measured in 2009, where they were looking at liquid jet with different concentrations of sodium hydroxide. And what they see is that you have the signature of hydroxide, which increases linearly with increase in hydroxide concentration. If it increases linearly, it does not show any evidence of an adsorption behavior. So it's just a bulk increase in concentration of hydroxide. And these measurements are also there were also some simulations by Pavel Jungwirth in the same group, in the same paper, where they show that, again, from MD simulations, there's no evidence of hydroxide directly going to the surface. So all of this together, we had to come up with a different strategy to look at this problem. So we wanted to directly measure oil droplets, not the air-water interface. And so our strategy was to make oil droplets in water, like I showed you before, and then measure the surface charge, then measure the interfacial structure of water, measure the interfacial structure of the oil at the surface layer, and hopefully there's a correlation between all those measurables. The first part is fairly simple, right? I can measure the zeta potential. Now, the zeta potential is not exactly the same as surface potential, but we won't get into that right now. It's just an indicator that it's negatively charged. And to measure the interfacial structure, we use this technique called some frequency scattering. So Sylvie Roque actually developed this technique starting in early 2003 or so. And so we wanted to use some frequency scattering to do these interfacial measurements. And so before I show you the some frequency scattering experiments, I want to show you the most or the classic example of a hydrophobic water interface, right? The most well-studied hydrophobic water interface is probably the air-water interface. So we can do the same SFG experiments I showed you in the first part, overlap the two beams and measure the reflected SFG beam. And the spectrum of interfacial water at the air-water interface looks like this. You have hydrogen bonded peaks, and then you have a sharp peak at high frequency. So for the hydrogen bonded region, we can, a simple way of interpreting the spectrum will be that you have a range of hydrogen bonding strength. Strongly hydrogen bonded water should show up at low frequencies, and weakly hydrogen bonded water should show up at higher frequencies. Now, if you have water molecules that are not hydrogen bonded at all, and this is the case for probably 10 or 13% of water molecules at the very top layer. These are the water that points their hydrogen towards air. So those hydrogens, those OH or OD in this case, because this is now measure of D2O water interface. So those OD groups, they are usually called free OD or dangling OD because they're not hydrogen bonded. They show up as a sharp peak around about 2700. So when we see the spectrum, we can say that, well, it's now let's do this measurement for the oil droplet surface and see if it looks exactly the same as the air-water interface. And it turned out it was actually more complex than that because we had a huge problem about the infrared having to pass through water, because for the reflection experiments, the infrared just has to touch the water surface. For some frequency scattering, the infrared beam has to go through water and reach the droplet surface. And in this process, it will get absorbed. So we first had to theoretically describe how this works. So we used some rather simple numerical simulations to understand how IR absorption is going to affect our some frequency response. So imagine this blue dashed line is our original some frequency response of water. And if I have an absorptive medium and the infrared is being absorbed, then after 100 microns or so, I'm going to measure a spectrum that looks like the green one. So there's quite a lot of spectral distortion because of IR absorption. So using these simulations, we came up with a way to correct for that and retrieve the true surface response in an absorptive medium. And so we first did that. And then we had to also demonstrate that this procedure actually works, not just for interfacial water. So we then went ahead and did the same type of experiment for the CH stretch vibrational region, which is a more crowded region. There are more peaks that are well ordered. And we showed that you can also use the same correction procedure to get interfacial spectra in absorptive media. So with these two studies, we had all the ingredients to go and do the measurement for oil droplets in water. Now, this is what that measurement looks like. You have interfacial water stretch spectrum next to oil droplets in water, and this is OD vibrations. So you have the spectrum going from 22 to 2800. It's just a broad, almost featureless spectrum. So we first compared it to the air-water interface to see what's really different. And if you look at these two dashed lines that I draw, which I call as strongly and weakly hydrogen bonded water populations, the ratio between those two peaks is different for the air-water interface and the oil-water interface. And we can actually calculate that ratio and then plot it as a function of temperature for the air-water interface. And this was done before. You can see that lower the temperature, higher the peak ratio. And that makes sense because when you go to low temperatures, hydrogen bonding network should get stronger. And now if you plot the ratio that we measured at room temperature for this emulsion, oil droplets, that ratio is higher than the air-water interface. So that is telling us that there is somewhat of a stronger hydrogen bonding network at the oil droplet surface compared to the air-water interface. Now, this is a very heavily debated area of interpreting the water spectra. What I gave you is the simplest explanation that we can give. But as I wrote here, vibrational coupling has a very strong influence on these spectral features. And by vibrational coupling, what I mean is that if you have every single OD is coupled to neighboring D2O molecules because they're all hydrogen bonded. So that is the intermolecular coupling. Intermolecular coupling can broaden the overall spectral width. Similarly, the OD vibrations within a water molecules can also couple with each other because the frequency of the stretch and the overtone of the bending of D2O, they exactly match. So you have a Fermi resonance between them that splits the OD vibration peak into two. So overall, coupling causes the peak to broaden, and coupling also causes the peaks to appear as double peaked. So we then had to do an isotopic dilution measurement where we dilute the D2O with H2O. And if you dilute it enough so that you go to 15% of D2O, so that is a black spectrum. Sorry, I think the legends are gone, but the 15% D2O looks like this. It's a much narrow peak, right? So you're essentially turning off the intermolecular and intramolecular coupling as much as possible. And then you see that the peak is now shifted. And I have a comparison of bulk water response here. And the bulk water response and the surface water response are shifted by around 12 wave numbers. And the surface water response has a shoulder around a low frequency shoulder that's absent in the bulk water response. So this isotopic dilution study was to really show that there is a stronger hydrogen bonding network, but overall it's not a very strong effect. You can see that the differences are quite they're quite small, but it's there because we have it verified with the isotopic dilution now. But the even bigger difference between these two spectra is this region, right? Where is the sharp peak in the oil droplet surface? Does this mean that there is no free OD at the oil droplet surface? There are no non-hydrogen bonded OD modes. So this is what we wanted to address. And if you look really closely at these two spectra, you see that there is a shoulder in the blue spectrum. It's not very obvious, but there's a shoulder. Is it possible that somehow this free OD then shifted to low frequency and merged together with the hydrogen bonded population? And this is what we wanted to check next. Now, if there is a shift, that would mean that some of the electron density from those interfacial OD bonds are being removed. And that's, if you remove the electron density, that should make the bonds vibrate at lower frequencies. So to verify that, we can use what is called as a polarimetric analysis. So all the measurements that I showed you were done with SSP polarization combination of the laser beams. That means that some frequency is S polarized or perpendicularly polarized. The visible is S polarized and IR is P polarized. Now, that's very important for the nonlinear optical signal that you measure. And then you can do the same measurement with a PPP polarization combination, and you take the ratio between them. So this is a ratio as a function of the infrared frequency. Now, this ratio should depend on the symmetry of the molecular vibrations that we are looking at. So for water molecules, we have mainly three types of symmetries. We can have a hydrogen bonded water molecule that's symmetrically stretching, C2V symmetric, or we can have an asymmetric C2V stretch, or we can have a free OD that's pointing towards air or pointing towards oil that will fall into a C infinity V symmetry. And these are the possibilities. And for all these symmetry groups, we can calculate using nonlinear scattering modeling, we can calculate what the intensity ratio should be. And then we see if our experimentally measured ratio matches with that symmetry prediction. And what we found out is that there is this region around 2600, it's quite broad, where we can match the experimental ratio with the C infinity V modes. So this tells us that there are free OD groups because C infinity V modes are the only ones that can correspond to a free OD mode. So you have free OD modes, but they are really shifted compared to the air-water interface. And that's giving rise to the shoulder. So as I said, a shift would mean you are removing some of the electron density from those interfacial OD bonds. So the question is, where is that electron density going to, right? And in this system, the only possible acceptor is the oil, because we have only oil and water. So we then had to look at the interfacial oil to see if some of that electron density is going into the interfacial oil bonds. So you can look at the CH stretch vibrations. And here we need a reference, right? And the reference is the oil droplet that's covered with a monolayer of lipids. And these lipids are deuterated DPPC lipids. So the point of covering it with the lipid monolayer is so that the oil or hexadecane is now no longer interacting directly with water because you have a lipid monolayer covering it. So these oil molecules are now sitting in a hydrophobic environment as if they are in a lipid membrane. And those CH modes vibrate at a frequency of around 2850. And then if you remove that lipid layer and you allow the oil modes to interact with water, you see that the CH2 vibration shift to higher frequencies. And that shift, again, as you can see, once you passivate the surface completely with DPPC, the surface, the zeta potential is close to zero. And the one that interacts directly with water has a zeta potential of around minus 50 millivolts. So the shifts also correlate with the overall charge that we observed. So as I said, now we have correlations that we see, right? We have the interfacial OD bond that shifted in one direction. We have the interfacial CH bonds that we shifted in another direction. So this when we put it all together, we came with the idea that we have charge transfer from the interfacial OD bonds to interfacial CH bond, interfacial CH of the oil. That's what makes the oil droplet surface negatively charged. And at this point, our theory collaborator, Dr. Ali Hassan Ali, was already working on this theory, and they had shown that using ab initio MD simulations, they calculated the charge density of individual layers, and they were able to calculate that there is a negative charge density that seeps into the oil. So this is the negative charge density in the oil at an, this was a hexane water interface. But interestingly, they calculated the surface charge density in the oil phase, and if we use something like a GUI-Chapman model to convert this surface charge density into a surface potential value, you get around minus 30 millivolts. That's fairly in the range of the zeta potential that we measure. So again, these theoretical simulations are confirming the idea of interfacial charge transfer, and the charge transfer occurs from these type of defects at the interface. So there are different type of hydrogen bonding defects at the interface, and those defects then transfer their charge into the oil phase. And while he was doing these simulations, he also saw an interesting observation that there are some interactions between water and oil that resemble a hydrogen bond. So these are not the typical water-water hydrogen bonds. These are much weaker hydrogen bonds. But if you look at this distribution of CHO angle versus CHO distance, you can see that there is a population that has angle less than 20 degrees and distance greater than 3.5 angstrom. And that can be classified as a weak hydrogen bond. And because this type of hydrogen bonds would result in a blue shift or a higher frequency shift of the CH. So we saw the higher blue shift in the CH peak from our some frequency spectra. So we then concluded that you do have some improper hydrogen bonds that form at this interface, and they mediate the charge transfer from interfacial water to oil. So with that, we now have a possible explanation for why oil droplets in water are negatively charged. We are able to show that we can measure the interfacial water and interfacial oil and then correlate those spectral shifts with the charge that we observed on the surface. So the charge really originates from the defects in the hydrogen bonding network of water. Now we could ask so many questions about how relevant are these for other hydrophobic interfaces. If I go to different length scale of hydrophobic interfaces, how these charge transfer effect could manifest. And these are all yet to be revealed. So with that, I would like to conclude my talk. Thank all these people, my advisor, Sylvie Roque, and the first part about metallomembranes was done in the lab of Professor Paul Kramer at Penn State. And then our hydrophobic interface work still continues with collaborations of Ali and Giuseppe from CNR. So we are working on understanding the pH-dependent effects of oil droplets that I showed you in the beginning. So the story is not yet complete from our side. So we are working on those with these two people. With that, I thank you all for coming, and thanks a lot for listening.
Thanks a lot for this beautiful talk. Now the room is open for questions. I don't know if there are questions here or online. For those of you online, you can use the Q&A option, and I'll track any question that shows up here. I can see that there are 2, if not 3 questions here. You were first. So
2 questions. One on the second part, and the other one on the first part.
Okay.
Okay, so you say that there is a charge transfer from water to the oil phase. How is that fits the surface tension graph that you showed, that when you add hydronium ion, it's the water goes, the surface tension goes down, and others it stays more or less the same, and with the hydroxide it goes up.
So I think these details are still to be understood. From the hydronium data, I think that at least the surface tension data gives you an idea that it might be going to the surface. Hydronium might be going to the surface. And then the question is, how does charge transfer, is charge transfer different when you have hydronium in the solution? I think this is not understood yet. For hydroxide, again, the same story. Right now, the simulations don't have any excess hydroxide in the solution. And same in our experiments, we are just keeping the water as it is. We are not adding anything extra to this. So it's not clear how charge transfer is going to be modified, but we do have some, as I said, with the collaboration with these two people, we then went in to look into the pH effects, and this is what we are working on right now. What we see is that the surface structure and the surface potential is not really changing when you go to high pH. We are not yet touching the proton yet, because the droplets are not stable when you go to low pH. So they start coalescing. They once the zeta potential gets lower and lower, they start coalescing. So we cannot do the nonlinear scattering measurements at that point. But for the high pH case, we see that there is really a discrepancy between what we measure as zeta potential versus what the actual surface potential is. So we think it's probably not really a surface phenomenon anymore. This is our working hypothesis.
Okay, and on the first project,
Yes.
you showed differences between zinc,
Yes,
and other bivalent ion. If we're thinking about ions, just a sphere with a charge, in your case, +2, all of them are just a sphere with a +2. How do you interpret the results that you get? The differences between the ions in your case are only the radius of these ions. So how do you interpret it? I mean, zinc, I'm not sure exactly the radius, but it seems to be somewhere in between the range of ions that you have, no?
Yes, and the radius, and therefore I think the most important will be then the hydration of the ion itself. So calcium versus magnesium, there's a huge difference in how they both interact. And because magnesium, if it has 6 water molecules in its first hydration shell, really likes to hold on to those water molecules. So, and that's also what we see in the measurement, that magnesium is not dehydrating the phosphate. So when magnesium binds, it's really binding with its solvation shell, so it forms a solvent shared pair. So I think the radius and the hydration hydration around these ions. Now, maybe that's not the full story, because for zinc, the way these ions coordinate to any specific ligands is different. For example, zinc, we show that it binds to phosphate and carboxylate, but all ions don't do that. So copper, for example, prefers to form at low concentrations. It prefers to form a square planar complex, not binding to the phosphate, just binding to the carboxylate and amine. So there are coordination preferences for these ions that might originate from their radius and hydration combination, I think.
Can you find monovalent ion with the proper radius that will reproduce the results of zinc? Or
So this is interesting. I have to look if anybody has done. People have definitely studied monovalent ions, but what we usually saw is that monovalent ions do not really. There were no monovalent ions that would dehydrate and form contact ion pairs. Now, lithium is small, but I haven't studied lithium, but probably, I don't know. I don't think this type of structural changes do happen with monovalent. They have been shown more with divalent ions. Yeah.
Marek, you have a
Yeah.
question?
Thank you very much for a very nice talk. It's nice to see this fundamental question called in interfaces sciences, which is still very relevant, but with time going, I think, lose the interest. So it's very nice to see how new techniques you can implement to solve this, answer these important questions. Regarding the first part, so that zinc can decrease critical micelle concentration of this phospholipid.
Critical micelle concentration, I would think yes.
I mean, I would expect that, right? If I have a micelle formation with this kind of phospholipid.
It's possible, yeah. So where are you getting with that?
I'm just asking to what extent it is. I mean, because you propose one type of ion with one type of phospholipid. So
Yes.
how universal it is?
So so for other lipids, right? I think this is something we have to test and see. I think in our case we tested PS and PG, another negatively charged lipid, and it was specific for PS and zinc. This interaction. But what you said about critical micelle concentration is true. So this could be more universal, but I don't think I can say I don't think zinc interacting with all the negatively charged lipids is doing this thing. So this is something we have to test and see. Yeah, but at least for the monolayer measurements, it will not affect the measurements itself, because what you're measuring is specifically surface and the surface density. Let's imagine that zinc binds to the lipids and brings some of them into the bulk. Once they're in the bulk, we don't expect them to change our interfacial signal, because at the interface, you're really compressing the monolayer. You're really compressing the monolayer constantly. So you're trying to make sure that you have the same density of lipids throughout your measurement. Yeah.
Okay. Yeah.
Is about the first part also, because at the very beginning you showed a table showing different ions, saying that the zinc is the one that dehydrates, and the other one not, but calcium
Also does, yeah.
Exactly, but was depending on concentration. And at the conclusions, I understood that all of these ions behave similarly, and zinc was the one different in the sense that it was below 30. That is the thing that you need to in order to dehydrate the lipids. But why calcium in higher concentration can also dehydrate the?
So we see a lot of similarities in the way the way calcium and zinc are binding, but calcium is much weak. It's a much weaker interaction. So if I compare 100 micromolar zinc with 10 millimolar calcium in spectroscopy, I see similar shifts happening. So then I conclude that, okay, zinc and calcium are both dehydrating the phosphate, but the strength of the interaction is quite weak for calcium. So what I think is that the way an ion binds, the structural details, where it binds, if it binds to phosphate or carboxylate, does it dehydrate the head group or not? These details matter. These details are specific to a concentration range, and that matters in how it restructures the membrane. So for example, if you just saturate with non-physiological, let's say 100 millimolar of all these ions, we haven't investigated what happens, but crazy things happen. So it's, I think whenever we talk about ion-specific effect, we have to, at least I try to stick to a concentration regime that's relevant for the binding constant of that ion. Yeah.
So I have two questions coming from the audience. I don't know. Okay. So A and B, I don't know who this person is. Maybe somebody does. Nevertheless, he or she is asking, for water around air bubbles,
Yes.
charge transfer would be rather difficult. In this case, would one expect indeed deprotonation of the interfacial water? Not my field. I'm not understanding well the question, but hopefully you do.
So I think they're asking about the other system. So we talked about oil droplets in water. Now they're talking about air bubbles in water, because I showed that both air bubbles in water and oil droplets in water have negative zeta potential. So in the case of oil droplets, we're arguing that there's a charge transfer from water to oil. Yeah. Now what happens for air bubbles? I think experimentally, I'm not ready to say yet because we haven't done measurements in air bubbles. But from Ali's simulations, he simulated not just oil-water interface, but also air-water interface. He sees that charge transfer is also present at the air-water interface. So it's, according to him, it's a more common phenomenon of a hydrophobic water interface. But then for the air-water interface, you have nothing to transfer the charge to. Right, so you probably have layers of opposite charges at the water surface. So he shows these charge density plots where the charge density oscillates as you go through the water layers. So at the air bubble surface, it depends on, I guess, what layer you're looking at. If you're looking at the very top layer, you do see a charge, but that charge is in the water itself. It's still residing in the water. Yeah, I think.
All right, and he or she has another question.
Okay.
It says, do the gauge defects cause broadening of the CH stretching SF resonances?
Okay.
I don't know if,
if
it's clear.
I understand.
And then they say, I found huge effect for amide terminated alkyl thiolated on gold.
So do gauge defects broaden the CH peaks? That's one. And then they found
a huge effect for the ¿cómo se dice amida en inglés?
Amide, amide.
Yeah, amide terminated alkyl thiolate on gold. Yeah, and A and B is Alex Bittner from.
I don't know. It would have been nice to talk to them in person if they're doing spectroscopy or
sure.
Yeah, but I think peak broadening, not very significantly that I have seen for SFG. So I think I have to be careful depending on what spectroscopy you're talking about. But in SFG, I showed at least this one plot where the CH2 peak was going down and the CH3 was going up. So this is the CH2 peak is what the gauge defects. If you have gauge defects, you see the CH2 peak. And generally, I think there is a small broadening, but it was not very significant. I did not quantify it. But I'm not sure about the correlation to the amide, and I think if I probably need more details to.
So I was thinking that maybe Alex, if you're still online, if you're interested, maybe you could meet this afternoon or.
Yes, yeah.
Nanoguna is right there,
yes.
The APC. If that's the case, Alex, please send me an email and I'll arrange something between both of you.
Yes.
All right.
Yes.
Okay. So I think with this, we can call this a seminar. So the discussion was very, very active and I thank you for that. And the talk was incredible. So thank you, Saranya.
Thank you. Yeah. Thank you all for showing up.
Thanks a lot. and stay in touch. Bye bye.
Bye.
All right.
Sí, los cuatro que había dicho.
¿De dónde Marimar? Qué maravilla, que ya no Pues sí.
Ah,
Hydrophobic.
yeah. Oh, you're doing surface force measurements?